And welcome back! Transcription makes 3 types of RNA from protein synthesis. Just like the last video, we'll run through these real quick. Messenger RNA carries the message that will be translated to form a protein. Blueprints for genetic coding. Ribosomal RNA, which is rRNA, forms part of the ribosomes where proteins are actually made. And transfer RNA, tRNA, brings amino acids from the cytoplasm to the ribosome to be matched with codons, which we'll talk about in the next slide. Next. Wait, next. Okay, there we go.
Now we're getting slap happy. Amino acids are coded by mRNA-based sequence. I thought you were laughing at me.
No.
Jeez. Okay, sorry. Translation converts mRNA messages into polypeptides. A codon is a sequence of 3 nucleotides that codes for an amino acid.
A polypeptide is basically a fancy way of saying an amino acid chain. And amino acid chain is going to be put together
To form a protein.
Yeah.
This protein is a bunch of chains of amino acids, or one chain of amino acids.
Okay, so why codons are so awesome? Everything is awesome!
Oh, the very bottom I kind of cut
You're
off.
part of the team.
Everything is awesome!
Okay,
Alright.
there are 64 different kinds of codons, but only 20 amino acids. So it is possible for an amino acid to be specified by more than one type or kind of codon.
This gives DNA a redundancy that helps prevent changes in the DNA sequence from actually affecting the protein that it makes. So that means if one little nucleotide gets changed on accident because DNA polymerase ain't doing its job right,
Oh, I'm gonna punch you.
It probably says Missouri.
it probably won't.
And crick.
It probably won't make a big deal. As you'll see in the next slide, when we show you all of the codons that code for like in a single amino acid, I'll kind of come back and show you what I mean by that.
Okay, so one codon specifies for one amino acid, meaning in a DNA strand with 12 nucleotides, it is going to have 4 codons. Those 4 codons will each code for a single amino acid. So at the end of translation, we would have a protein with 4 amino acids long.
Yes.
A protein made out of 4 amino
Yeah, made out of 4 amino acids.
Yeah.
So one codon equals 1 amino acid.
Yeah, so there's the chain of DNA. TAC, HAG, GTA, CAG. Those are 4 codons.
Here, they can't see you pointing.
I don't know where it's at. Okay, there.
So this
Those
was
are
the
DNA.
gibberish she just said.
Codons, and then it goes to RNA,
This is
codons.
RNA.
and so
Would be your mRNA.
yeah. Well, I just said RNA. But
That's okay. I just
yeah,
was
mRNA,
specifying.
and then it goes to
The tRNA is going to come in and bring in these specific
yeah.
amino acids, and it's going to link them together to create a polypeptide chain.
So just
Yep.
remember, if you have 4 codons, you're going to have 4 amino acids. If you have 3 codons, you're going to have 3 amino acids.
Well, if you want to say it's simple, yeah.
Yes, it's 1 to 1.
Yeah.
Why would
yeah.
we do that? We're science
Mhm.
teachers. Okay, the genetic code matches each codon to its amino acid or function. Okay, so here you go. There's 3 different stop codons. Where's the pen? Okay, do we actually... Alright, so here's a stop codon. Oh, jeez. Maybe you should do this.
It's okay.
This is good.
And then there's
And then the third one here. So there's 3 different codons that tell RNA polymerase stop. And there's one start codon. Let's see.
Isn't
an
it
AUG. Where's...
right there?
Okay, oh, it doesn't say start.
No, it doesn't.
Okay, but that's your start codon. Okay, oh, it does. Codes for...
right there.
Okay,
So you have 3 that tell you to stop and 1 that tells you to start.
yep.
That's as simple
Pretty
as you
much
need to
it,
do. Yep.
Alright, a change in order in the order in which codons are read changes the resulting proteins. What does that mean? Okay, it's called a frame shift, right? Are we getting into that?
Just not too
specific.
They just
yet?
have to know.
Okay, so this. See, a change in the order in which codons is going to change the protein. They've got an example in there. Do you want to draw, or someone want to draw? Because, oh no.
Draw what?
I don't know what you're...
Go through and show the shift. So we start here with this.
So you have CGA up here, right?
That's where we originally would start.
If you want to back up the video, you can see what amino acid that would code for. And just a little shift, meaning if we took out this C, oh, there we go. Arg. Alright. So if the C gets switched up or maybe deleted, however the frame shift happens, okay, it will completely change all of this. So now instead of starting with arg, we have asp, which is GAU. Alright? So it can change the entire protein. That's why frame shift mutations are very dangerous and very bad.
Yeah, it's going to have a domino effect on the amino acids that are strung together.
And we'll go over mutations next week.
Okay, someone else want to do this one? Oh, geez, oh, geez.
Okay, amino acids are linked to become a protein. An anticodon is a set of 3 nucleotides that is complementary to an mRNA codon. An anticodon is carried by tRNA. So you're going to have an amino acid, you'll have tRNA that is carrying all of this, and then your anticodon is going to be the base pairs that match up to the ones that go to the codon.
Yep.
Okay, for translation to begin, tRNA. I don't know why that's such a tongue twister to me.
tRNA?
Yes.
I can just, every time we come to it, I'll pop and say.
Sub for me.
Yeah.
It binds to a start codon and signals for the ribosomes to assemble. So it's like a whistle or something that's getting blown that tells them to begin. Sorry, I'm all about analogies.
Do you want me to say the next one?
Yes, go ahead.
Okay, a complementary tRNA molecule, see that's how I say it, just kind of rolls off the tongue.
And not to be.
tRNA molecule binds to the exposed codon, okay, bringing its amino acid close to the first amino acid. So we have this ribosome, as you can see in the picture here. Okay, it's pointing here, all that green stuff, that's ribosome. Okay, is going to come in and it's going to connect onto, this is actually your mRNA, which I don't know if we really need to go that in detail.
No.
Okay, that's your messenger RNA that's telling basically the ribosomes what amino acids they need to put together. Okay, so the tRNA, it's kind of like a little car that's bringing in this amino acid, and it's going to bring it in here, and then it's going to get attached to this one, creating an amino acid
It's kind of like a drive-through bank. They run through the ribosome, each tRNA holding its own amino acid, parks when it matches up with the mRNA, and releases its amino acid, and it gets bind to the other one left behind. So all these cars are going through, and a chain of amino acid is being created out the back here. Okay?
And you can see over here that it shows longer ones that have been created because the tRNA has already been through.
Those are rich people banks.
We don't live there.
The ribosome helps form a polypeptide bond between the amino acids. The ribosome pulls the mRNA strand the length of one codon. So that's just going to tell you,
3 nitrogen bases.
Yeah.
And then it moves to another 3, and another 3. Obviously, because it's going codon by codon.
Because it has 3 anti.
Yeah.
Okay. Yeah.
Yeah.
Well, that picture also shows that there's space in the ribosome for more than just one tRNA. So they kind of line up and have to wait for a little bit to release their amino acid.
Until they get the right TNA. It's spreading!
See?
Alright, tRNA that has the correct match for these down here.
The codon
Once
and the
we,
mRNA. Yep.
yeah, once we have a correct match between the codon and the mRNA and in the tRNA, then the
amino acid is released.
yeah, we know we have the correct amino acid, unless there's been some kind of mutation. And we got the correct.
Which we'll get to later.
Yeah, we got the correct amino acid, so we can build proteins.
Yeah.
Okay.
The now empty tRNA molecule exits the ribosome. A complementary tRNA molecule binds to the next exposed codon. Once the stop codon is reached, the ribosome releases the protein and disassembles because it knows it's done.
It's probably why we call it the stop codon, so it will
Tell it to stop.
tell it to stop.
And again, the stop codon's like another whistle being blown to tell them to stop. It's enough.
Hey, guess what? I think we've just reached a UG.
Stop.
Stop!
Right.
All right, you're done.